All right, moving forward to the next section, we have Claire Dacombe, Vice President APAC from Asia Bioseparations, where she will talk about reliable solutions for downstream processing contaminant removal. So before we start, just a brief background of Claire. Claire is based in Singapore with over 10 years of experience managing the Asia-Pacific commercial market for products focused on cell line development, biomanufacturing, and bioprocess. In her role for Asia Bioseparations as the Vice President in Asia-Pacific, she is developing regional bioprocessing market with a resin, nanofiber, and services portfolio to support customers with their bioprocessing needs. Asia Bioseparations are developing scalable, reliable solutions with resin used in more than 20 FDA or EMA-approved manufacturing processes. So now I'll pass the time to Claire for her presentation. Please welcome.
Thank you. Right, good morning everyone. So I'm really pleased to join you live here, and today we're going to talk about downstream processing for contaminant removal. So I don't really need to cover much of this because I was already introduced, but basically I'm based here in Singapore. I've been here for coming up to 11 years now. When I started, I was working as a global application specialist, mainly focused on cell line, so upstream processing, and recently transitioning to downstream processing, so working under Gamma Biosciences, of which Australia Bioseparations is one of the operating companies. So just to give you a brief overview of Gamma Biosciences, in case you're not familiar, this company has a group of operating companies focused from discovery all the way to kind of downstream processing and manufacturing for advanced therapies. So whether this be biologics, vaccines, and the cell and gene therapy space. So in the discovery and cell engineering space, we've got Mirus for our transfection and expression reagents, as well as biomagnetic solutions for cell screening and selection. In the bioproduction space, we have Universal Technologies. So this is our fixed bed reactors for high production of viral particles and vaccine particles, as well as Nirin Technologies for those inline analytics. And then in the bioseparations, so downstream processing space, we have Australia Bioseparations, which I'll be focusing on today, for the chromatography, resins, and bioprocessing columns, as well as Nanoparel for our kind of novel nanofiber-based chromatography membrane. And that's going to become really critical as we go into advanced therapies more for the purification of larger modalities, so things like virus particles, plasmid DNA, messenger RNA. But as I said, today we're focusing on Australia Bioseparations. So this company's actually been around for quite some time. It's over 30 years old, but you might be more familiar with the name Prometic Biosciences. We have very large-scale manufacturing, so we can make individual batches of up to 1,000 litres. This is all done in clean room manufacturing, and so we can provide you with the regulatory support files that you need in order to submit your products into clinical trial. So as you can see, we have various products that have already gone through FDA approval or the equivalent regulatory bodies. This is a timeline slide, so I'm not going to go into it in detail because I know these are never particularly interesting. But the critical things are, in the late 80s, we were a spin out from the University of Cambridge in the United Kingdom. In the late 90s, they developed their proprietary resin, so it's called the Purabead, and I'm going to cover that a little bit more in a couple of slides. There were various product developments and manufacturing developments over the years until in 2019, Australia was acquired by Gamma Biosciences. And actually, it was in 2020 that it was rebranded to what we are now, which is Australia Bioseparations. We do have a global presence. I've been with the company just under a year with the prospect of kind of developing the Asia-Pacific market. So we have now set up various distributors here. So we've got a strong network to support the market here, as well as direct presence, including key territories of China and Japan. So that's the introduction to the company. So the first thing I wanted to cover was our base matrix. So as I mentioned, we have our own proprietary base matrix that we manufacture in-house, which is called the Purabead. And this is an agarose base matrix. And one of the unique things is in the beading process, so the manufacturing of that bead, we don't use organic solvents or toxic materials like oils, so it makes the manufacturing a much greener process. One of the other things you get from this beading process is you get very uniform bead size. So you can see, oh, press the wrong button. So you can see here the size distribution for Pura bead is very consistent. Whereas if you compare this to an alternative market-leading product, you see this very large distribution in the bead size. Why is that important? Well, when you're packing your columns, you want to have a nice uniform packed column, like you see in this diagram here. That's much easier to achieve when your beads are all a consistent size. If you have this broad size distribution, you will get this non-uniform packing. You might even find a gradient of the smaller beads at the bottom, the larger beads at the top. And this is going to impact your application. And so here you can see this is one of the impacts that you'll see with the Pura bead. As the pressure increases, you can see the flow rate increases in a pretty much linear fashion. If you compare this to an alternative bead on the market, you see that the flow reduces. And so this is going to impact how you run your applications, but also you'll have the implication of back pressure. Some of the other key features, security of supply. I don't think any of us are unaware of the challenges we've had following the pandemic with supply chain. Well, because we manufacture this in-house, and as I said, it's based on agarose, not cephorose. We don't have any of those challenges of supply. We manufacture to 9001 and can offer you the regulatory support files. I mentioned the large batch sizes. And of course, as we're all looking to reduce our environmental impact, we can help to support you to generate greener manufacturing processes. So this table just gives examples of the off-the-shelf products that we have. So down this side is just alphabetical order. So more interesting is probably along the top, which is application-based. So we have affinity-based resins for albumin and albumin fusion proteins, for alkaline phosphatases. We also have products specifically for antibodies and antibody fragments. So our Fabsorbent is for your antibody fragments, and we have Mabsorbent for those IgGs or monoclonal antibodies. So this is kind of an alternative to protein A. It doesn't function in exactly the same way, but it gives a much more cost-effective solution versus protein A. And it can also bind IgG3. So if you're looking for that isoform, protein A is not able to bind that. This binds all isoforms. We have some general purification, and I'll talk a little bit about our Hicks later in a case study. So we have our Hicks and ion exchangers. And then when it comes to impurity removal, we have an endotoxin removal called Atoxiclear, which I'll cover a little bit. We have IsoClear for removal of your isoglutenins, and we have PABA for the removal of serine proteases. And then one of our other off-the-shelf products, which seems to be gaining a lot of traction at the moment, is our insulin and insulin analog resin. So this is an affinity-based resin for the purification of those products. So the first case study I wanted to talk about was the hydrophobic interaction. This you may be familiar with. It's a very versatile purification technique. And typically what you might do is you might screen a number of ligands and identify which one is best suited to your product and your process. So at Astrea, we offer screening kits. Oh God, I keep pressing the wrong button. We offer screening kits, so butyl, hexyl, octyl, and phenyl. And you can then run the elutions and identify which one is best suited to your product. So what you can see on this side is the elution patterns of the 4 different Hicks. And based on that, you can then decide which one is most suitable for your process. And so in this case study, what they were aiming to achieve was to remove the dimer aggregates. So in this case, the dimers are a contaminant to their final product. And so you can see the first thing they did was do a screen of all 4 Hicks, and within a gradient elution. So the idea is to identify which is the best Hick for this removal of dimers, and at which salt concentration are we seeing the best elution of the monomer. And what you can see is this top band, the higher molecular weight, that's the dimer, and the larger band below, that's the monomer. And so if you look at these 4 plots, you can see that actually only phenyl is able to elute without eluting the dimer as well. So that indicates phenyl would be the Hick of choice in this application. The next step then is to do the experiment in a way that's more like you would in a true downstream processing, which is using a step elution, so changing the salt concentration as a single step. And so here they've selected a concentration which showed the best elution in the gradient, done the step elution, and you can see the results that even with the step elution, there's less than 1% of the dimer aggregate present in the elution. So this is a nice example of using Hicks to remove a contaminant. The next one I wanted to cover was endotoxin. I'm sure probably everyone in the room is familiar with endotoxin, particularly if you're doing downstream processing. So this is a lipopolysaccharide. It's produced by gram-negative bacteria. It's toxic. So if you use an expression system like E. coli, you expect to have this burden which you need to clear. However, you can see this burden in pretty much any expression systems. And the levels of endotoxin you can have in your final product are negligible. So what we've developed at Astrea Bioseparations is an affinity-based ligand on a resin for removal of endotoxin. So this next slide, you can see our in silico team modeled the lipopolysaccharide. You can see there are various binding sites on it. It's particularly challenging because it's very pH stable and it's very temperature stable. As I said, if you're working with E. coli, you would expect contamination that you need to clear, but we do see unexpected spikes in endotoxin late in a bioprocessing, so you don't want to lose that batch. So we want to be able to ensure that that batch isn't lost even if you see this. And, you know, there is a kind of move towards more efficient downstream processing, which is obviously looking at reducing the number of steps that we take in downstream processing. The challenge here is we can then fail to address this endotoxin burden. So what we have is we have a specifically designed ligand for endotoxin removal. What's unique about this is it's very low protein binding, so you recover your target proteins. It's manufactured under GMP environment, so it's ISO 9001, and we have all of the regulatory support files. And you can see we offer this in various pre-packed sizes as well. So what I wanted to do was show you some of the data of the clearance from Etoxiclear. So on the left-hand side, you can see a chart which is comparing Etoxiclear with a couple of other of the standard endotoxin removal membranes that are on the market. Obviously, the advantage is this is a resin. When you are using a membrane, there is a challenge with scale-up. You could see a challenge with fouling. But really what's noticeable here is if you use the ion exchange endotoxin membrane, and this would be applicable if you were using an ion exchange resin as well, you are clearing the endotoxin to less than the detection level of this assay, but you're losing a significant amount of your protein. Whereas if you look at this affinity-based endotoxin removal, you're maintaining your protein recovery, but you're not actually clearing the endotoxin to the level that's required. So Etoxiclear is kind of in this sweet spot in that it can remove your endotoxin and you can also recover your protein. So on the right-hand side, we just wanted to validate that this is applicable to both low endotoxin burden and a very high endotoxin burden. And what you can see with the final endotoxin concentrations is that it works in all those scenarios. Those were kind of bench scale. What the next thing is going into process scale. So as we scale up, does this still work in the same way? And so this is an IgG which had low or high endotoxin, so you can see across the top. And we loaded it into a 50 ml Evolve R, so pre-packed column. Have one of those on the exhibition stand if you want to see what it looks like. And then we scaled that up into our larger 7 centimeter Evolve D column, which we also have on the exhibition stand if you want to see it. And that's about 385 mils. And what you can see is in all cases, we're recovering over 98.5% of the IgG, and we're also clearing more than 4.5 log of the endotoxin. So with the pre-packed columns, we offer this Etoxiclear in a variety of pre-packed from the 7 centimeters all the way to the 20 centimeter diameter columns, and from bed heights of 5 to 20. And these are all validated. They have the regulatory files with them. And we actually do offer those Evolve D columns for any of our resins. So all of those resins that I showed you on the table, you can get them pre-packed in these Evolve D columns. What that can mean is they're kind of plug and play ready to use. So it can save a lot of time and money. You don't need the expertise in-house to pack, to QC, and then to provide the validatory files to go with that. So we can offer that for any of our resins. However, you may be interested in packing your own bioprocess columns. So we do offer these larger bioprocess columns. They go from 7 centimeters and now actually up to 65 centimeters. And we're continuing to scale these up and develop larger columns. They conform with all the necessary requirements. They have very high performance. So you're rated up to 4 bar for the pressure. One of the critical things is these can actually be very cost effective. The reason being, you buy a column, you don't have to replace that whole column when it's time to change your column. We have what's called a refresh kit. So any component in the column that's been in contact with the wetted path, you can just change that part of the column. And that means that it's only 1/3 of the cost of the column versus buying a complete column. So in the long run, it becomes a very costly way of doing this. But also, it is another way to promote greener manufacturing, right? There's much less waste if you're not replacing the whole column each time. So that was a very brief overview. So hopefully I've shown you that we can offer you security of supply for both resins and columns. And actually for the bioprocessing columns, I should say, we have these available. So if you order them, they're delivered in a matter of weeks, because we do understand that at the moment with the supply chain challenges, columns can be pretty challenging to get hold of. We are present in all regions globally, and we've really beefed up our presence in Asia Pacific to be able to offer you the support that you need here. And we have a variety of solutions for optimizing your biologics purification and polishing. And with Gamma as a complete portfolio, we really can help you from discovery all the way through to downstream processing and manufacturing for advanced therapies, and of course the cell and gene therapy space as well. Thank you. And if there's any questions.
Thank you very much, Claire.
No problem.
All right. So we have some questions from the online first.
Yeah.
Okay. It asks, can ectoxic clear handle turbid feed streams? How clear should the feed solution be at the inlet?
For the atoxic clear?
Yes.
So we have never seen a challenge with the fouling or non-specific binding. So I don't think I think you can use it pretty much straight out of bioreactor. The feed doesn't need to have pre-polishing to be able to use it. It can be used as an initial step, or it can be used as a final step.
Thank you very much. And we have one more question from our online site. Can ectoxic clear be used for endotoxin removal in viral applications, such as AAV?
Yeah, that's a good question, actually. So it can be used for AAV, endotoxin removal, but as we know, resins maybe aren't best suited for larger modalities. This is where the nanofiber membranes come in. So what we are at the moment developing is that endotoxin removal onto the nanofibers, which is the nanoparel side of the business. And this will then really allow you to do that in the most streamlined way. But currently, yes, people do use the resins for the removal of endotoxin in viral applications.
All right. Also, before we move on to the next question I have here, do we have any from the on-site attendees? All right, no problem. We'll move on to the next one here. Sorry, Claire, we have one more question here. She said, you mentioned that the HIC resins are available in small columns.
Yeah.
What sizes are these?
So we have them in pre-packed 1 mil and 5 mil columns for process development. And again, if anyone wants to see what they look like, they are on the exhibition stand.
Thank you very much, Claire. And once again, thank you very much for your presentation. So again, if you are interested in Asia bioseparation strategies and also some solutions they provide, please feel free to check out their booth at the exhibition hall to discuss more opportunities and also collaboration with them.